I am Lavanya. I am from JSS College, Department of Chemistry. Today I am here to representing a seminar on affinity chromatography. So first we discussed about the chromatography. What is chromatography? It is a collective term for the set of laboratory techniques for the separation of mixtures. Basically, chromatography is known as the separation techniques used in the laboratory. Then we discussed about affinity chromatography. It is a purification of a biomolecule with respect to the specific binding of that biomolecule due to the chemical structure. Affinity chromatography means purification of biomolecules with respect to the specific binding of the biomolecule due to its chemical structure. It is known as the purification technique. Affinity chromatography means the purification technique in that we separate the biomolecules. Then it is a method of separating of mixture of proteins or nucleic acids by specific interactions of those molecules with a component known as ligand, which is immobilized on a support. If a solution of, say, a mixture of a protein is passed over the column, one of the proteins binds to the ligand on the basis of a specifically and high affinity. The other proteins in the solution washed through the column because they were not able to bind to the ligand. So the other solutions means the impurities are washed out because they were not able to move. So they were washed out. Example, enzyme with an inhibitor, antigen with an antibody, etc. So this is the affinity chromatography diagram, which is explained the lock and key type mechanism. So first the matrix and the ligand combine to form a immobilized ligand. The immobilized ligand means the solid bound to the complex. The immobilized ligand and the sample combine to form complex. Then the impurities were go outside. Then the complex will detached the complex and the purified samples are separated. So we discussed about the principles. First one is affinity chromatography is one of the most diverse and powerful chromatographic methods for the purification of a specific molecule or a growth of molecules from the complex mixtures. Then it is based on highly specific biological interactions between two molecules, such as interaction between enzymes and substrate, then receptors and ligands, or antibody and antigen. Basically, the affinity chromatography means it is highly specific biological interactions. Then it is occurs between two molecules, so such as interaction between enzymes and molecules, enzymes and substrates, receptors and ligands, etc. These interactions, which are typically reversible, are used for the purification by placing one of the interacting molecules, referred to as affinity ligand, onto a solid matrix to create a stationary phase while a target molecule is in the mobile phase. Basically, the target molecule is in the mobile phase. Many of the commonly used ligands coupled to affinity matrices are now commercially available and are ready to used. Then another one is the separation of proteins or biomolecules by the reversible interaction between a protein or a group of proteins and a specific ligand coupled to a chromatographic matrix. So these are some types of affinity chromatography. The first one is lectin affinity chromatography, then immuno affinity chromatography, metal chelate chromatography, diligand chromatography, then covalent chromatography. So let us know about chromatographic media. So basically, chromatographic media means it is known as a chromatographic resins or adsorbents. So then a matrix in its use here a substance usually in bead form to which specific ligand is covalently bound. The substances usually they are in bead form to which the specific ligand is covalently bound. In order to for the matrix to be effective, it must have certain characters. The certain characters are it must be insoluble in the solvents and buffer. employed in the process. The certain characters are the some of the characters are the solvents, the substances must be insoluble in solvents and the buffer solutions. So then next one is it must be chemically and mechanically stable. That should be chemically and mechanically stable. Then it must be easily coupled to a ligand or spacer arms onto which the ligand can be attached. Then it must exhibit good flow properties and have a relatively large surface area for attachment. It must have a good flow properties. Then the process will be occurs very easily. So next, what is immobilized ligand? Basically, the immobilized ligand means the immobilized ligand means a molecule that has been chemically bound to a solid support like a column or a sensor surface. The ligand can be selected only after the nature of a macromolecule and the isolated is known. When a hormone receptor protein is to be purified by affinity chromatography, the hormone itself is an ideal candidate for the ligand. The hormone itself is an ideal candidate. Then, for antibody isolation, an antigen or hapten may be used as the ligand. Antibody isolation In antibody isolation, the antigen or hapten may be used as the main ligand. If an enzyme is to be purified, a substrate, analog, inhibitor, cofactor, effect, or may be used as the immobilized ligand. So let us know about the attachment of a ligand to the matrix. Several procedures have been developed for the covalent attachment of the ligand to the stationary phase. All procedures for gel modification proceed into two separate chemical steps. The two chemical steps are activation of the functional group on the matrix and the joining of the ligand to the functional group on the matrix. The first one is the functional group activation. Then second one is joining of the ligand to the functional group on the matrices. Then a wide variety of activated gels is now commercially available. The most widely used are described in the following. So specificity is based on three aspects of the affinity. The first one is matrix, spacer arms, and the ligand. Matrix for ligand attachment, spacer arm used to bind the ligand to the matrix. The ligand molecule that binds reversibly to a specific target molecule. That's the site of interaction. Then the examples are antigen to antibody, antibody to antigen, substrate to enzyme, DNA to histone, hormone to binding proteins or the receptors. So let us know about advantages of affinity chromatographies. The first one is it is highly specificity. Then the target molecules can be obtained in a highly pure state. Then it is a single step purification, so the time duration is very low. Then the matrix can be reused rapidly. So that matrix is cannot be used. It is used reused. Then the matrix is a solid. It can be easily washed and dried. So the next one is give purified product with high yield. Affinity chromatography can also be used to remove specific contaminants such as proteases. Then the disadvantages of the affinity chromatography. In this affinity chromatography, we use expensive ligands, then leakage of the ligand, then degradation of the solid support, limited lifetime, non-specific adsorption, then it is relatively low productivity. So by this picture, we know how the sample introduction takes place and how the elution is done. So here the sample is there. Then first affinity ligands are covalently attached to a chromatography support and packed into a column. Then the sample is flowed through the affinity column in a binding buffer. Then the target molecules bind to the affinity ligands. Then the sample is washed through the column and remove the unbound components. Then the elution buffer is applied to the disrupt the interaction between the affinity ligand and the target molecule. Then last, the purified target molecule is eluted from the column. So this is the last topic in the affinity chromatography, that is applications. The first one is it is used for the isolation and purification of all biological macromolecules. Then it is used to purify nucleic acids, antibodies, enzymes, and etc. Then to notice which biological compound bind to a particular substance. So it is used for the some of the biological compounds, how they are bind to the particular substance, that we can easily observe in this affinity chromatography. To reduce a amount of substance in a mixture used in genetic engineering, nucleic acid purification. It is used in genetic engineering also. Then production of vaccines, antibody purification from the blood serum. So this is used in vaccines and antibody purifications in some blood samples, etc. Then and basic metabolic research, proteins or enzyme purification from cell-free extracts. So by this, I conclude this topic. The first so by this introduction and by this presentation, we know about how affinity chromatography works in biological chemical interactions and principles, applications, then the ligand matrix attachments. So this is the final conclusions. So thank you.